Now we are going to discuss another nephrotic syndrome, that is membranous nephropathy. That is called as membranous nephropathy. Most common cause of nephrotic syndrome in elderly patient. Remember, this is the most common cause of nephrotic syndrome. Most common cause of nephrotic syndrome in elderly patient, right? So in elderly patient, this will be the most common cause of nephrotic syndrome. Can be a future question, right? When we talk about membranous nephropathy, we should understand membranous glomerulonephropathy, they consist of two subtypes, primary and secondary. Primary is idiopathic, where it is unknown, right? And remember, 70 to 80% cases are the primary only, right? So 70 to 80% cases means majority of the cases. So that what I wanted to tell that most commonly membranous is the idiopathic. So membranous is most commonly idiopathic and very rarely they are secondary, right? So secondary is less common, right? So I want to say that secondary is less common. Secondary is secondary to what? Secondary is secondary to drugs like non-steroidal agents, NSAIDs, right? It can be secondary to infections. Secondary to infections. Which infection? It can be hepatitis infections, right? So secondary to hepatitis, it can be secondary to infections like hepatitis. In hepatitis, we all know that there are several types of hepatitis B, A, C, right? So here I would like to tell you about hepatitis B and hepatitis C. Two important things, right? So for hepatitis B, what is the most common glomerulopathy? So if examiner says in hepatitis B, which is the most common, right? So in hepatitis, most common, when we are talking about hepatitis B, most common in pediatric age group. In pediatric age group is the membranous glomerulonephritis, and in adult age group, it is MPGN, right? So if examiner is asking about adult MPGN, pediatric age group, it is the membranous. And what is overall? So that pediatric one is the overall also, right? So this pediatric is the overall also. So overall also it is membranous glomerulopathy, which is most common, right? So it is membranous glomerulopathy which is most common overall also when we are talking about pediatric membranous adult MPGN. What is the most common association with hepatitis C? This was previous year All India question also. Hepatitis C is most commonly associated with, remember, hepatitis C is most commonly associated with cryoglobulinemia. So they are most commonly associated with cryoglobulinemia. After this, membranous glomerulonephropathy, and the last one is the MPGN type 1. So this is the order which we need to remember, that hepatitis C is most commonly associated with cryoglobulinemia. This was the All India question previous year, right? If it is not given, think about membranous, then MPGN type 1, right? But when we are talking about hepatitis B infection, so hepatitis B infection is most commonly associated with membranous. When pediatric, it is membranous. When it is adult, it is MPGN, right? Two other infections which are most commonly associated with membranous is the syphilis and schistosomiasis. These were All India question in 2013. Syphilis is most commonly associated with, so syphilis and schistosomiasis, right? So syphilis and schistosomiasis both are most commonly associated with membranous glomerulonephritis, right? So both are most commonly associated with membranous glomerulonephritis. So syphilis and schistosomiasis both are most commonly associated with membranous type of glomerulonephritis. So that is the another important point which we have to keep in mind for our exam point of view, right? Now coming to the next thing in this is membranous glomerulonephritis is also associated with, third is cancers, right? So they are associated with cancers, or you can say malignancies, right? So it is associated with cancer, breast, cancer, colon, right? So cancer colon was the question in All India 2014, right? So this was the question in All India exam 2014. Cancer colon is most commonly associated with. Cancer colon is most commonly associated with. Answer was membranous glomerulonephritis, right? So right now we are studying about membranous. So cancers are or malignancies, right? And that was the question in AIMS 2015. Malignancies are most commonly associated with, right? So these were the questions in All India exam 2000. See, this was the question in All India exam 2014, and this was the question in AIMS exam 2015, right? Malignancies or cancers are most commonly associated with membranous. Here what we need to be cautious in exam time. If examiner says lymphoma and leukemia are most commonly associated, remember this point. What I am talking about solid cancers, right? When we talk about cancer colon, breast cancer, then you have to think about membranous. But when examiner talks about leukemia and lymphoma, then you have to think about minimal change disease, as we have discussed just now, right? So these are important point about membranous glomerulonephritis etiologies, right? Now we will see what is the pathogenesis of membranous glomerulonephritis. Pathogenesis of membranous glomerulonephritis can be due to immune complexes, right? So these are basically due to immune complexes, right? So immune complexes are responsible for the damage, right? Immune complexes are responsible for the damage because these immune complexes will be releasing cytokines, and these cytokines will be causing damage to kidney, right? So this will be causing damage to the basement membrane, as well as visceral epithelial cells, right? So here also we are seeing same like a minimal change disease, but here it will be basement membrane will be affected, there will be damage, right? So that is the pathology behind this membranous glomerulonephritis. What we need to remember extra here, immune complexes which we see in this patient is usually IgG4. So this type of immunoglobulin complex will be present, or IgG4 deposits will be characteristic of the deposits seen in membranous glomerulonephritis. This can be a new question from the neurobins, right? So immune complexes are IgG4 type of deposits which we may find in case of the membranous glomerulonephritis, right? So as I said, basement membrane and visceral epithelial cells both will be having damage. So what are these damage? In basement membrane, they will be having thickening of the basement membrane, right? In visceral epithelial cells, there will be same thing, effacement of the foot processes of visceral epithelial cell, right? So these are the important findings in the pathogenesis which you will see in case of the membranous glomerulonephritis patient, right? Membranous glomerulonephritis, right? Here what are the interesting point we need to understand in their pathogenesis when it was conducted on a mice, right? So antigen used was mice proximal tubule antigen. So proximal tubule antigen of mouse was used for experimental model of the membranous glomerulonephritis. That was the All India question, right? So remember, that was the All India question. Megalin antigen is the antigen which had been used for the experimental model of the membranous glomerulonephritis. Remember, experimental model of membranous glomerulonephritis, right? And this type of nephritis is called as Hayman's nephritis, right? So that is another important MCQ in exam. What is Hayman's nephritis? It is the experimental model of membranous glomerulonephritis, right? So megalin antigen is responsible for the damage, and this damage will be causing Hayman's nephritis. What is Hayman's nephritis? This was nothing. This is the membranous part of the glomerulonephritis, right? So both are same thing, right? It was an experiment of the mouse, and this is seen in normal human being, right? And this is seen in human being, right? And this is seen in human being. right? This is one thing. Second thing, corresponding to the megalin antigen, because megalin was present in mouse. So what is present in human being? What is present in human being? In human being, you will find they are having phospholipase A2 receptor. So this antigen is corresponding to the megalin. So remember, this phospholipase A2 receptor is corresponding to the megalin antigen present in mouse, right? So basically it is immune complex mediated, where antibodies are formed against phospholipase A2 receptor, and phospholipase A2 receptor is working like an antigen for membranous glomerulonephritis, right? So in human body, antibodies are targeting phospholipase A2 receptor, which is acting like an antigen, and the concentration of this antibody against this phospholipase A2 receptor, this is the biomarker of the disease activity. Remember, this antibody against phospholipase A2 receptor, when you will check them inside this serum, right, they will be considered as biomarker of disease activity. Biomarker of disease activity. So what is biomarker of disease activity? So biomarker of disease activity is the phospholipase A2 receptor antigen, right? So this point we have to keep in mind, right? This can be a future question, and this was the question in DNB exam previous year. Phospholipase A2 receptor is associated with membranous glomerulonephritis, right? So membranous glomerulonephritis, we have seen they are associated with the antigen called megalin in mouse, phospholipase A2 receptor in human being, and antibodies are formed against them, immune complexes are formed, and when cytokines are released, they will cause damage of both visceral epithelial cells and the basement membrane, right? So now we are going to see those things on microscopic examination. So when you are doing light microscopy of membranous glomerulonephritis, what finding you are going to see? So there are two important findings. Number one, you will find capillary basement membrane thickening. Capillary basement membrane will be having thickening because of the deposition of the immune complexes, right? So capillary basement membrane thickening because of the immune complex deposition. What we need to remember, there will be no rupture of the basement membrane. This was the All India question in 2016. No rupture of the basement membrane. So there will be no rupture of the basement membrane will be seen, right? So there will be no rupture of the basement membrane in case of the membranous glomerulonephritis. So capillary basement membrane thickening, and second important point, there will be no rupture of the basement membrane. That is the light microscopy, right? And if you see, they are having immunofluorescence, right? Immune complex mediated disorder. So what should be the immunofluorescence microscopy here? So if you're looking at the immunofluorescence microscopy, suppose I am showing you these are the glomerular capillaries, and this is the one in large view. You will notice that they are immune complex mediated, so they will be forming granules, right? So they will be forming granules, and this is called granular immunofluorescence. This is called granular immunofluorescence. So granular immunofluorescence are present because it is an immune complex mediated disorder. Because it is a immune complex mediated disorder, right? So granular immunofluorescence, immune complex mediated disorder is there, and that is why they are having this kind of presentation, right? So that is the immunofluorescence microscopy. Coming to the electron microscopic examination, what are the findings you will see on electron microscopy? On electron microscopy, you will see that there is a capillary, there is a basement membrane, right? As I have told you, capillaries are lined by endothelium, endothelial line, right? These are the basement membrane, and here I would like to tell you, number one finding is the visceral epithelial cell, right? So because of the cytokine release, there will be effacement of the food process of the visceral epithelial cell. So number one thing which you will notice here, effacement of food process of visceral epithelial cells, right? So effacement of visceral epithelial cells will be seen on microscopic examination, electron microscopic examination. Second thing, what we are going to notice that they are having phospholipase A2 receptor on their basement membrane surface. See what I am showing you, all these are, this is hypothetical diagram, right? This is just hypothetical diagram. What I am writing Y shape is all are, these are all phospholipase A2 receptor. So these are all phospholipase A2 receptor which is going to behave like an antigen, right? So what I'm showing you on the basement membrane, phospholipase A2 receptor is there, on which there will be antibody deposition. So there will be antibody which will be getting deposited, right? So now we can see this is the antibody which is getting deposited. So this antibody antigen complexes, they will be forming immune complexes, right? They will be forming immune complexes like this, right? So they will be formed between the membranous area also and above the basement membrane, membranous area also and above the basement membrane. So what I'm showing you, these are the immune complexes, right? So these are the immune complexes which is partly on the basement membrane surface because of the presence of the phospholipase A2 receptor antigen, and partially above just below the foot process of the podocytes, right? So that is how we are going to see a membranous glomerulonephritis. So because of immune complex deposition, what we are thinking here, what is happening, there is a thickening of the basement membrane. There is a thickening of the basement membrane. Remember what I have said in earlier light microscopy, thickening will be there but there will be no rupture. Thickening but no rupture of the basement membrane. Thickening of the basement membrane, but there is no rupture of the basement membrane. That is one important thing we have to understand, right? So there is a thickening of the basement membrane but no rupture. Second thing, you can see the deposit present in the membrane also, present above the membrane also, but below the food process. So these are also called as subepithelial deposit. So number 3 finding you will notice here. So this was the second point. Third is the subepithelial deposits. Right, so immune complex deposits are subepithelial deposits. These subepithelial deposit, if you look, it appears they are above the basement membrane, right? Because partly in the basement membrane and partly above the basement membrane below the food process, right? So that is why it is also called as epimembranous deposit, which was all India question. Epimembranous deposits are seen in, right? So that is why they are also called as epimembranous deposit, older all India question. Epimembranous deposits are seen in membranous glomerulonephritis. When you are seeing this kind of deposit, and if you observe what you are observing here, between the deposit there is a portion of basement membrane, right? So there is a portion of the basement membrane. So immune complex is like a dome, right? So immune complex is like a dome and basement membrane is like a spike, right? So that is why it is also called as a spike and dome pattern. A spike and dome appearance is diagnostic finding on electron microscopy of membranous glomerulonephritis, where a spike is made up of basement membrane and dome is made up of immune complexes, right? So now we can decipher very easily the content of dome will be corresponding to the severity of the membranous glomerulonephritis, right? So we can say like this, कि dome will correspond, means size of dome will correspond to the severity, right? And what is the size of dome? Now the question is, what is the size of dome? Dome is immune complex. And what is immune complex? Immune complex is having phospholipase A2 receptor antigen, right? So phospholipase A2 receptor antigen is the antigen which is going to form immune complexes, right? So now we are seeing immune complexes are formed which is corresponding to the severity of the membranous glomerulonephritis. So that is why when we were discussing in the pathogenesis, I said okay, serum phospholipase A2 receptor concentration is biomarker of the disease severity, right? So now we can correlate these things together, right? Look at the microscopic view. When you are seeing electron microscopy, look at this electron microscopy. in this electron microscopy, this was the previous year AIMS question also, you can observe that this is the basement membrane, right? This is the lumen and this is the endothelium, right? So now we are seeing endothelial cell, nucleus, lumen, basement membrane. In the basement membrane, you can observe there are dark gray area and light gray area, right? Dark gray area and light gray area, right? So what I want to show that this dark gray area is the immune complex which is forming dome, right? And this area which is remaining between dark area is called as basement membrane, which is forming a spike. So this is how spike and dome appearance will be visible, right? So now you can see that you can easily make it out here also. There is a immune complex, right? This is the basement membrane area, and again you can see there is a immune complex. Again there is a immune complex, and in between there is a basement membrane. So this is called spike and dome appearance. Examiner may ask this question: spike and dome appearance is best appreciated by which microscopy? So remember, it is best appreciated by microscopy, not by electron microscopy. It is best seen by light microscopy, best seen by light microscopy with special stains, with special stains, like pass or silver, right? Pass or silver. Pass stain will show red basement membrane, silver will show black basement membrane. So that is another important point we need to understand. Spike and dome is best seen by light microscopy with special stain called silver stain or pass stain, right? So immune complex is there, and that is why when you will see the immunofluorescence microscopy of membranous glomerulonephritis will be granular, right? So that is why they will be having granular immunofluorescence because of immune complex mediated pathology. And when you will see the light microscopy with special stain, this is the pass stain, and this in this inset is called silver stain. So these are the two stain, light microscopy with two special stain. Pass stain, you can make it out. These are the spike and there is a dome, right? So spike and dome, right? So this is the spike. Spike is the basement membrane. So basement membrane is red, dome is light color. So it's a spike and dome pattern, right? So easily you can make it out a beautiful spike and dome here on light microscopy, right? Same way here you can see the dark color, the black color is the basement membrane, and the red color area is the immune complexes, right? So this is the silver stain. So spike and dome is best visualized on light microscopy with pass stain, where basement membrane is looking deep red, and on silver stain where basement membrane is looking darker or blackish. What is the clinical presentation of this patient? When you will see clinical presentation of the membranous glomerulonephritis, patient will be usually, it can affect all age group, but usually they are adults, right? And as we already discussed, they are the most common cause of nephrotic syndrome in elderly, right? This patient, most common presentation is the massive proteinuria. So most common presentation is the massive proteinuria. Most common presentation is the massive proteinuria, right? And remember, this massive proteinuria will be non-selective. So minimal change disease, it was selective, and here it is non-selective massive proteinuria, right? Non-selective massive proteinuria, plus this patient may have, I'm not saying always, this patient may have hypertension and hematuria. So this patient may have hypertension or hematuria. So these are the important presentation of the patient clinically. So they will be presenting with the massive proteinuria, and they will be having hematuria, they may have hypertension, and as I have already told you, bioactivity, disease activity can be, biomarker of disease activity can be measured by phospholipase A2 receptor antigen, and antibodies formed against them.